Good day everyone. I am Dr.K.SELVANAYAKI, Department of Microbiology. Today we can see about monoclonal antibody. These monoclonal antibodies are very helpful in the current scenario of clinical treatments and various aspects of immunology. The monoclonal antibodies are antibodies which are targeted about the single epitope. That many epitope, if it is targeted, means that polyclonal antibody, but the group of cells which are produce the antibodies against a single epitope means that is called monoclonal antibody. The monoclonal antibody is first formulated by George and Caesar in 1975. So what they did is they have formulated the two cells. One is a myeloma cells, another one is a plasma cell. The plasma cells are generated by B cells. So the plasma cells are generated when the antigen is getting antibodies against the antigen. Now for the formulation of monoclonal antibody, what they are doing is they will inject the antigen, particular antigen, into the mice or laboratory animals, so that the B cells will encounter that particular antigen and they will undergo many cells. That is, the cells will be formulated, it will get multiply and differentiated into two memory cells. The plasma cells will give continuous production of antibodies. That antibodies are targeted the antigen. So now the monoclonal antibodies are targeted about the single epitope. Next cell is a myeloma cell, that is a cancerous cells. What the character is, the character of the myeloma cells are cancer cells which undergo continuous cell division. So when the two cells are combined, this will give the hybridoma cells, get fused inside the Sanday virus or inside the polyethylene glycol. So when the two cells are together, it will give the hybridoma cells. That the hybridoma cells may be 2-3 types or hybridomas may be produced. So unfused cells may be there for plasma cell and the myeloma cell. Some cells are having two nucleus, both are belong to plasma cell, either belong to plasma cell, or two nucleus either belong to myeloma cells. Some of the cells only generate myeloma cells. So now we have to go for identify the hybridoma cells. For that they are undergo hat selection. The hat selection is nothing but the what is meant by hat means H refers for the hypoxanthine and A stands for aminopterin and T stands for thymidine kinase. So these three combinations, when the three combinations are present in that media, that is used for the hybridoma action. So these cells are grown into the hat medium. So normally the cells are either produced by undergoing cell division. So the cells are, before going for cell division, they have to get ready for their nucleus. That means replication, DNA replication it has to undergo. Usually the cells will either follow de novo pathway or salvage pathway. The de novo pathway, so here the phosphoribosyl pyrophosphate and the uridylate is present in the de novo pathway. Both will be produce the nucleotide and the DNA will be formulated. In this case, the hat selection, we are block the de novo pathway, so that the pathway is blocked by the incorporation of aminopterin in that media. So the cells no other way to go, they have to undergo salvage pathway, they have to select the salvage pathway. Here we are taking the plasma cell and myeloma cell. Why we are means the plasma cells will continuously produce the antibodies. Here the myeloma cells will continuously undergo cell division. These two characters are very important. In the same way, they are hat selection is based on the HGPRT, that is hypoxanthine guanine pyrophosphate and thymidine kinase. Here the media which is incorporated with the HGPRT, the cells are we are selecting the cells which are positive for HGPRT and thymidine kinase for undergo salvage pathway. Here the plasma cells are HGPRT positive, thymidine kinase negative. In case of myeloma cell, they are HGPRT is negative and the thymidine kinase is positive. When these two cells are combined together, they will only grow in the hat medium. If it is get fused together, the two nucleus belong to the plasma cell and the myeloma cell one present means that can only grow in the hat medium. So that salvage pathway to undergo cell division in the presence of DNA replication. So, salvage pathway means the cells must contain positive for hypoxanthine and thymidine in the presence of HGPRT and thymidine kinase. So, the cells only grow, the hybridoma cells can grow in the HAT medium. Based on that, the monoclonal antibodies are produced in for various type of. For example, in the cancer treatment, the cancer cells are targeted and we have to load the drug which will kill the cancer cells. So, the drug is uploaded or the drug is pinned with the monoclonal antibody. The monoclonal antibody will travel towards the epitope of the cancer cell only. It is targeted only to the cancer cells, so that it will go and bind to the cancer cell. The monoclonal antibody will go and bind to the cancer cells and it will release the drug on the cancer cells, so that the cancer cells will be destroyed completely. So, then we can sort out sites, B cells, T cells, and then many type of cells are targeted based on the count. Cell count can be achieved by monoclonal antibodies. So, various identification and including treatment, we are using the monoclonal antibody. We can see elaborately about the applications in the next video. Thank you.